Hi, today we're going to go over chapter 24 from Frederick H. Martini, the digestive system. Our goals are to identify the major organs of the digestive system and their function. Here is an overview of the digestive system. You're going to have a muscular digestive tract and several accessory organs whose role is to promote and aid in digestion. These are the functions of the digestive system. You have the initial ingestions, the mechanical processing, followed by a chemical digestion, eventual secretion. Between these, we also have absorption and excretion. By secretion, we refer to the secretion of enzymes that aid in the digestion and eventually breaking nutrients and macromolecules down enough for to their elemental particles in order for us to digest them. The digestive organs are housed within the peritoneal cavity. The peritoneal cavity is surrounded by the serous peritoneum, which contains two layers, a visceral and a parietal layer. Um, You also have mesenteries joining different regions of the digestive organs. And we'll see embryologically we had an anterior and posterior mesentery. The posterior mesentery is the one that houses or joins several of the digestive organs, whether the anterior mesentery. We can find that in portions of the lesser omentum and in portions of the falciform ligament. Here we have that view of the developing of the mesenteries, where we had a ventral and a dorsal mesentery. The ventral mesentery, as you can see, eventually loses most of its adhesion to the digestive tract, but it remains here, contact joining the liver to the anterior wall, and this is done through the falciform ligament. Here's a view of that ventral mesentery joining the liver and the stomach through the lesser omentum. But most of the other mesenteries are going to be part of the embryological dorsal mesentery. Just a note to um, a factoid about the development of these mesenteries. Here you have a view of the abdominal cavity with all of the peritoneal organs removed. As you can see, there are certain organs which are retroperitoneal. They're behind this peritoneum. That includes the kidney. That includes the pancreas. You see the duodenum. It's also retroperitoneal at this level, and you will see also the rectum following through. Here is a view of the peritoneal cavity and all of the digestive organs being lined by being surrounded by this mesentery. As we notice, most of the dorsal mesenteries are going to be the one surrounding these most of the digestive organs, and you have portions of the ventral mesentery lining adhering the liver to anterior cavity to the falciform, the anterior portion to the falciform ligament, and also the lesser omentum at this view. You have here also the greater omentum present here, the greater omentum and the lesser omentum are these pads of adipose tissue that's used for protection as well as storage of nutrients. Whenever there are times in which we store a fair amount of nutrients and you start seeing this portion of adipose tissue grow and grow and grow and grow, especially during quarantine or during your first year of freshman year. We're gonna focus now on the histological organization of the digestive tract. For the most part, um, you will see a mucosa, you will see a submucosa, a muscularis externa, and either a serosa or an adventitia. The mucosa, it contains mucous epithelium. It's moistened by glandular secretion, and it also contains a lamina propria. This lamina propria will provide vascularization to the mucosa. This is areolar tissue, and you will also see in certain portions you'll see a muscularis mucosa, mucosal layer, muscular layer within the mucosa. The submucosa is composed of dense irregular connective tissue. It contains vascularization as well as um, this submucosal plexus, which is just innervation that aids in the expression of components of the submucosa, especially glands. This is important considering that these glands are usually there to prepare the digestive system through the passage of food. So we need that vascularization of the autonomic nervous system, letting these glands allowing these glands to prepare the lumen of the digestive system for the incoming bolus of food or the incoming chyme. Here you observe the muscularis externa, and this will be a smooth muscle. You usually have a circular and longitudinal. In the stomach, you will also have an oblique layer. The muscularis externa will contain a myenteric plexus that will aid in the activation of smooth muscle. This is especially true during peristaltic movements. Your serosa that surrounds this tract provides protection as well addition to surrounding area. These are the layers that we just referred to. So you will have present your mucosa, and your mucosa will contain your epithelium and this areolar connective tissue here, layer depicted in yellow here. You will also have a muscularis mucosa, and this is depicted here. Your submucosa containing dense irregular connective tissue. In this case, you observe a submucosal gland present here, and you observe that it is highly vascularized, as well as you will observe these plexuses, these submucosal plexuses, um, um, aiding in the function of the submucosa. Here you have a view of the muscularis externa. Your inner layer is your circular layer, and your outer layer is your longitudinal layer. As we talked about during the lab, in order for peristaltic movements to occur in this lumen, first you squeeze, and it makes sense that the circular layer is the first one whenever you look at it from the perspective of the lumen. And furthermore, you start this peristaltic movement following the contraction of these longitudinal muscles. Here we observe this is a view of the mesenteries that line all of these digestive organs. They're highly vascularized. We also observe a view here of this specialization that increase the surface area by 600-fold of the epithelial layer of the digestive system. This increase in surface area aids in absorption of nutrients. So here you see this plica circularis, this especially these infoldings of the submucosal layers allow this plica to form. You also see this villi present at the surface, and we zoom in further, you will see that each one of these villi is composed of a layer of columnar simple columnar epithelium, and you will see that the surface of the villi will contain microvilli, further increasing the surface area. Here we observe the movement of digestive materials. This is an example of peristalsis. Once again, there is an initial contraction of the circular muscles, and eventually you observe longitudinal muscles alongside a sustained circular contraction of the circular muscles propelling this bolus of food. Um, this at the moment, whenever ingestion happens and the initial mechanical digestion occurs at the level of the oral cavity, this is known as a bolus. Eventually, once it reaches the stomach, it will be known further along as chyme whenever true digestion occurs. The digestive system will be controlled by neural mechanisms. These are the mechanisms that are under control of the parasympathetic nervous system, mechanisms that we're not uh, aware of or that we don't control. We don't control whenever a peristaltic movement occurs or whenever digestion occurs. You also have hormonal mechanisms which will aid in the aid in peristaltic movements, as well as it will be involving certain levels of hunger or satiety, such as the case of leptin and ghrelin. So you would also we also have local mechanisms that, for example, distension of the stomach or changes in pH. They can serve as a chemical stimuli in order for um, for a response to occur. In this case, some of the response would be secretory cells releasing buffers, acids, enzymes, and so forth. This is a view of all of these mechanisms. We mentioned about neuronal control. Stress receptors can, through the myenteric plexus, can um, affect the central nervous system. And from there on, you can have this output, peristaltic, and so forth. Mm. Let's look at the oral cavity. I'm going to breeze through the oral cavity since a lot of this is anatomy mostly. But this is wherever mechanical processing occurs. There's also a level of lubrication and limited digestion. Here you have components of the oral cavity. Once again, we will further focus on this during the lab. The primary functions, mechanical processing. um, you will also have there's within the tongue you will have analysis of touch, temperature, and taste receptors. You would also have at this level secretion of mucins and lingual lipase by the tongue. This will help lubricate the food and initiate the digestion of lipids. Here we have a view of the salivary glands. You have your parotid, sublingual, and submandibular. Your parotid secretes salivary amylase, which aids in the digestion of carbohydrates. Sublingual secretes mucus, and the submandibular secretes both mucus and salivary amylase. You have about 1 to 1.5 liters of saliva secreted each day. Most of it produced by the submandibular gland, followed by the parotid, and eventually the sublingual. The saliva itself contains electrolytes, buffers, glycoproteins, antibodies, and enzymes. Their function includes lubrication and initiation of digestion. Here is a view of the salivary glands. Here you observe these two different types of glands present here, mucus cells and serous cells. You have mucus cells secreting these mucus substances, while serous cells secretes either digestive or buffers or anything that would aid in the digestion of food. So here you will have ducts. This is where all of these different acini, all of these different compartments, these different circular compartments, they eventually merge into bigger and bigger ducts, and those ducts will lead to the surface of the oral cavity. Here you have your parotid salivary gland, and through the parotid duct, they will reach the oral cavity, submandibular gland to the submandibular duct, and you have here your lingual, sublingual gland, and several submandibular ducts. Your teeth will focus mostly in the lab, but they aid in mastication, have three layers, enamel, dentin, and root. Um, Here you have, this is a view of your primary and your secondary teeth. These are your baby teeth, and here you have whenever you grow into this secondary teeth. Let's look at the pharynx. So this is the common passage of food, liquids, and air. It contains, due to this amount of abrasion, it contains stratified squamous epithelium. Pharyngeal muscles are going to assist in swallowing. These include the pharyngeal constrictor muscles and your palatal muscles. Once the food gets transferred from the oral cavity through the pharynx to um, the esophagus will be responsible for passing food from towards the stomach through the cardiac sphincter. So here you have the different layers of the esophagus. You have this due to the amount of abrasion that occurs, you have a stratified squamous epithelial layer present, and in this view you observe the mucosa, these dark blue areas, you saw mucosa present here, and your muscularis. Let's look at the process of swallowing. It has three different phases: the buccal, pharyngeal, and esophageal phase. Um, so here you have your buccal phase aided by the tongue. Your pharyngeal phase is going to involve the the um, the elevation of the larynx in order for the epiglottis to cover the opening towards the larynx and the trachea, and eventually you have here at the level of the esophagus at the initial portions you have this skeletal muscles that are going to aid in the initial swallowing event. That is why we are in control of when we swallow. Eventually, once it passes the esophagus, it's going to pass through the diaphragm, eventually through the cardiac sphincter into the stomach. Let's look at the stomach. Functions of the stomach include the storage of undigested food, mechanical breakdown of food, and disruption of chemical bonds via acids, and the production of intrinsic factor, which will help with the vitamin B12 absorption, important in hematological processes. At this point, whenever acids from the stomach mix with the food bolus, you will the food bolus will be known as chyme at this level. Notice that digestion occurs, but not much absorption occurs within the stomach. This is due to the fact that we have the acidity of the stomach. We have a protected layer of mucus to prevent damage of the epithelial layers uh, to the acidity of damaged epithelial layers. I'm gonna pause this recording because there is somebody mowing. Nope, I reckon we continue. Okay, so I hope I'm recording. Okay, I'm gonna continue. So here's the anatomy of the stomach. Here let me we're gonna focus on the cardia, the fundus, the body, your pyloric sphincter, and your rugae. Your pyloric sphincter and your rugae. So here we have that view of the stomach. This is mostly anatomy, but here's the fundus, the body, your pyloric sphincter. You have your ruga, and here you have the cardia the cardia, and within it we will observe the cardiac sphincter. Here's a view of the histology of the stomach. It contains simple columnar epithelium. This simple columnar epithelium produces this carpet of alkaline mucus, which protects the epithelial layer of the stomach from acidity. Um, Gastric glands contain parietal cells and chief cells. Parietal cells will secrete intrinsic factor, while chief cells produce hydrochloric acid. Parietal chief cells are going to produce pepsinogen, which is a proenzyme which will get activated into pepsin with the stomach pH. Pepsin will allow the digestion of proteins. We also, within the pyloric glands, you will also observe enteroendocrine cells, G cells that secrete gastrin, which stimulate parietal and chief cells, and they also increase gastric contractions, which are the part of this mechanical processing. And you also have D cells, which continuously secrete somatostatin, which inhibits this gastrin, the release of gastrin. It basically it inhibits digestive functions. We also have other hormones of notice. You have ghrelin, whose increase leads to hunger. So if it's 6 p.m. You haven't had dinner, you have ghrelin increasing, and you will have this context of hunger. Leptin, on the other hand, as you are enjoying a meal, leptin will increase until we feel the feeling of satiety. This is ghrelin is released by adipose tissue. Here is the view of the simple columnar epithelium, the surface of at the surface of the stomach, and here are these gastric pits, these gastric glands. So, what's the definition of a gland? Um, it's a cell and organs that release into a duct that gets exposed to the surface. In this case, here is the gland, and what are we releasing? We're releasing hydrochloric acid. We're releasing pepsinogen. We're just releasing gastrin into these pits, into these glands. Here you have a view of the glands themselves, and here you have your parietal cells, chief cells, and at the bottom you will have your G cells. These are all part of the mucosa. So here you have your mucosa. Here's your muscular mucosa. Your lamina propria is kind of embedded within the gastric glands. But this is a view of the surface of the stomach. So here are the glands themselves. These pores that you observe here, they are going to be the glands. Here's an schematic view of what we're referring to. So here you have this layer of simple columnar epithelium that is connected to these glands. And within these glands, you have your parietal cells, chief cells, and G cells, all part of the mucosa. This is a view of the excretion of hydrochloric acid by the parietal cells in a way that it's not is not detrimental to the cell or its immediate environment. So what we observe here is chlorine and this hydrogen ion being released separately by different mechanisms. So we have here CO2 and water leading to carbonic acid, and this carbonic acid dissociates into a hydrogen and bicarbonate. The hydrogen gets transported to the lumen of the stomach, while simultaneously we have a process where the bicarbonate, through an antiporter, moves chlorine from the extracellular environment into the lumen into these parietal cells and then eventually into the lumen of the stomach. So now you have HCl, which have high affinity towards each other, forming this highly acidic hydrochloric acid. The regulation of gastric activity occurs through a cephalic, gastric, and intestinal phase. Let's look at what this entails. So here is the cephalic phase. The cephalic phase. During the cephalic phase, we have input from the from the central nervous system. You have here vagus nerve number 10, promoting mucus release, release of pacinogen, HCl, and release of gastrin. So this is the end result. We are getting prepared for a meal, and through the release of these substances, will, whenever the meal arrives, will be ready to initiate digestion. The gastric phase it initiates by this mechanical input by the bolus of food, which at this level will be known as chyme, by the chyme of food distending the stomach, and in turn, this gastric portion will lead to the release of more gastrin. Gastrin will in turn feed back into promoting further release of these enzymes and acids that promote the breakdown of pepsinogen, promote the breakdown of proteins, and HCl in turn the breakdown of all components. This the initial the initiators of this response are going to be stress receptors and chemoreceptors that notice these changes occurring at this level. Gastrin itself is also going to promote the peristaltic movements within the stomach, which leads to an increase in mechanical digestion. We also have an intestinal phase. This intestinal phase, what it does is once the chyme reaches the intestine, it inhibits, it falls back into these components of the within the stomach and inhibits the further digestion, as well as it increases the peristalsis, inhibits peristalsis as well, and this induces the release of substances that aid in digestion of these substances that have reached now the intestinal portion. Digestion and absorption in the stomach occurs through salivary amylase, pepsin, and lingual lipase. So we have here three enzymes that are able to digest carbohydrate, proteins, and lipids respectively. Absorption is limited due to the mucus coat, due to the fact that gastric epithelium lacks transporter for these macromolecules. The gastric lining itself is impermeable to water due to that mucus coat that we have present, and digestion is not complete. We still have we still have molecules that are pretty much not broken. And we need the molecules broken down almost to their elemental and like to their monosaccharides, to the amino acid level, to the lipid fatty acid level in order for us to initiate digestion. Ethyl alcohol can diffuse through the mucous barrier. That is why if you drink within a meal, you ethyl alcohol, you will be able to feel it rather promptly. Now let's move on to the small intestine. 90% of digestion occurs within the small intestine. It's aided by secretions by the pancreas, liver, and gallbladder. We have three divisions within the small intestine. You have the duodenum, jejunum, and ileum. The longest being the ileum, followed by the jejunum, where most digestion and absorption occurs. The duodenum can be considered the mixing bulb, and this is because it receives inputs from stomach, pancreas, and liver and gallbladder. We mentioned that this portion was retroperitoneal. Eventually, once the chyme makes it through the small intestine, it reaches the large intestine through the ileocecal sphincter. Here we have a view of the small intestine, the duodenum, the jejunum, and here we have the ileum leading into the cecum of the small intestine, of the large intestine. Histology of the large intestine, we have these specializations, the plica, these folds of the intestinal lining, which occur, these are folds of the submucosa and everything that follows it. You have villi, microvilli, and villi, you have here several villi present, and you have within the surface of this villi, you will see microvilli, which is known as a brush border, all of which increase surface area. You will also have the presence of lacteals, so each one of these individual villi will have components of the lymphatic system, and this the purpose of these lacteals is to aid in the absorption of lipid substances or fatty acids, which will eventually get bundled into these micelles that allow these micelles, these balls of protein surrounding lipid substances, that allow the transport through the hydrophilic blood and lymphatic system and lymphatic vessels, lymph itself. Here we have the intestinal wall. Here we have an example of a plica circularis and composed of several villi. You see here your mucosa, your submucosa, your muscularis externa, and a serosa. This is a view of an individual villi. Here we will be able to observe the brush borders. These are the microvilli present in the surface of each one of these simple coronal epithelial cells. You will also observe these mucous cells present here. Um, and at the core of each villi, you will have a lacteal that aids in the absorption of lipids. So as nutrients pass through this simple columnar epithelial layer, lipids will eventually enter the lacteals. And other nutrients will enter this highly vascularized capillary area. Here we have the intestinal secretions. Their function is to moisten the chyme, help buffer acids, and aid with digestion. Enzyme secretion is going to be done through, we have brush border enzymes which activate trypsinogen. This is a pancreatic proenzyme. So it is these enzymes, what they're aiding is aiding in the activation of enzymes that have reached the duodenum. You also have these other cells, enteroendocrine cells producing cholecystokinin, gastric inhibiting peptide, and secreting. Duodenal glands with themselves produce large amounts of acids large amount of mucus in order to protect the duodenum from the incoming chyme. The chyme just came from the highly acidic stomach, and this alkaline mucus, has protected the duodenum. So here we have some of the intestinal movement, peristaltics, segmentation, and you have these other gastroenteric and gastroiliac reflexes. Let's look at the function of the pancreas. The pancreatic duct enters the duodenal wall. It has an endocrine function, which involves the secretion of insulin and glucagon, and it has an exocrine function. Um, the release of pancreatic juices will contain water, ions, and enzymes. These enzymes will be these enzymes are carbohydrates, lipases, nucleases, and proteolytic enzymes. Most of them are going to be proteolytic enzymes. And basically, the enzymes released by your pancreas will aid in the digestion of proteins, nucleic acids, lipids, and carbohydrates. Here we have this view of the enzyme. The enzyme, this exocrine function of the enzyme involves the secretion of components. We mentioned these pancreatic enzymes into ducts. So you observe a lot of these as cini present within the pancreas. These are basically simple cuboidal epithelial cells that secrete their components into these lumens. These lumens go into bigger and bigger ducts, and eventually those ducts enter the pancreatic duct. And from there, the pancreatic duct meets the common bile duct, and they release through the sphincter of Oddi, or the hepatic pancreatic sphincter into the duodenum. This is where you have stomach components, and you have components from the liver, the bile composed by the liver, and you have all of these enzymes composed by the pancreas are going to mix at the duodenum. Let's look at the liver. The liver performs metabolic and hematological regulation. And the liver, it's basically the site where it determines whether the level of nutrients are adequate for the rest of the body. If there are too many nutrients, the liver can store some of these nutrients in the form of glycogen. But if they have not enough nutrients, the liver can this catabolize this glycogen and start to produce glucose that will be freely available within the circulation. The histological organization is organized in lobules. These are hexagonal in shape, and in each corner it contains triads. What's a triad? You have your hepatic portal vein bringing nutrients from the digestive system. You have your hepatic artery proper, just bringing oxygen to the liver, and you have the bile duct leaving the liver into the duodenum or the gallbladder if it needs storage. The lobules contain a single cell thick plate of hepatocytes. Basically, the lobules are disposed on these single layers that are in contact with these sinusoids. The sinusoids are these openings through which blood can flow. And so, what we want is high contact between hepatocytes and the blood from the digestive system in order for this regulation to occur. And this is where it happens. These exposed hepatocytes are exposed to the surface of the liver. They contain microvilli. This is a view of the portal of how the circulation within the liver occurs. So you have your portal vein and hepatic artery blood percolating through the sinusoids, eventually reaching the central vein. From the central vein, it goes to the hepatic vein, inferior vena cava, and eventually the right atrium. So basically, you take components coming from the digestive system and oxygen percolates through the sinusoids and then eventually return this blood that has been regulated by the liver for nutrients, and it returns through the inferior vena cava with deoxygenated blood. Ducts, on the other hand, this is you have ducts in a similar fashion by going in the opposite direction. You have bile canalicula, bile ductus, bile ducts, which uh, form into the right and left hepatic ducts, eventually into common hepatic ducts and common bile and cystic ducts. Liver function is involves the regulation of nutrients and toxins in the blood. So you have carbohydrate metabolism, lipid metabolisms, all of this being regulated. It regulates levels of triglycerides, regulates amino acids, removing excess amino acids, removes waste, in neutralizing ammonia into urea. It stores vitamins, minerals as well. And your hematological regulation involves this part of the immune role through Kupffer cells. These are antigen presenting cells, which are basically surveying what we have just digested, that's coming through the portal veins, what we have just digested, and searching for foreign invaders. You have synthesis of plasma proteins, albumins, which this is the main site of plasma protein synthesis, and you also have the synthesis and secretion of bile. But let's start looking at this is anatomy. We'll focus on that on the lab. This is a view that we can focus on. Here we observe this is a portal triad. This is a, a hepatic globule. All of these individual cells are hepatocytes present here, and you see them. These are present in these thin layers, in these thin channels. Within the lumen of these channels contains these sinusoids through which blood can percolate. So you have here a portal triad at the corner of each of this hexagonal lobule, and you have a central vein present here. Blood through the portal vein is coming from the digestive system, and also blood from the hepatic arteries are coming. This is blood coming from the heart. This is oxygenated blood. This blood percolates through the sinusoids and gets in contact with the hepatocytes. The hepatocytes here are regulating for everything that we mentioned: amino acids, lipids, carbohydrates, and eventually, once that regulation occurs, all of this blood will reach the central vein which will lead to the hepatic artery proper, then eventually to the um, inferior vena cava and the heart. So this flow going from the portal triad into the central vein, there's an opposite flow occurring from the hepatocyte into the bile. These bile ducts present in the portal triad. It's done through these different levels of complexity of bile canalicula, bile ductules, and eventually bile ducts. So all of the bile that's being formed by the hepatocytes is being collected, and eventually we reach the bile duct. The role of bile is acts as an emulsifier. It basically breaks down lipids into smaller droppers so they're easier to digest and absorb, and it also promotes the actual absorption of lipids. Here we have a view of the gallbladder. So bile is being produced and eventually being collected into left and right hepatic duct. This comes through the common hepatic duct, and if a meal is not we haven't had a meal, this bile goes through the cystic duct into the gallbladder for storage. And whenever a meal is ready, we release components of the gallbladder or bile from the gallbladder through this sphincter into the duodenum. So let's look at the coordination of secretion and absorption. So we have the secretion of all of these enzymes that we need for digestion to occur, and we also have to coordinate the absorption in order for this to occur so by the time they reach the absorptive surfaces of the jejunum, that we have nutrients that are fairly digested. So this is a view. Food enters the stomach, and then we have here production of acids and motility, also some enzymes. Eventually, this chyme enters the small intestine, which releases gastric inhibitory peptide, which falls back and decreases some of these stomach functions. It also secreting and cholecystokinase is going to induce the release of the pancreas of all of these buffers and hormones that aid in the digestion of food and are released into the duodenum. Let's look at the large intestine. The large intestine's role is the reabsorption of waters, the absorption of vitamins produced by bacteria, and the storage of feces. You have different. We have a column with four different structures. You have the ascending, the transverse, and the descending, and the sigmoid column. We also have these specializations called the cecum and as well as the rectum. The column will contain these pouches called hastra. It also contains this tinea coli, which are portions of smooth muscle, which aid in peristaltic movement. Um, what else? Here from the ileum, it has to pass the ileocecal sphincter in order to components from the ileum to reach the cecum. The rectum, this is a large portion of the digestive system. It terminates in the anal canal and it has internal and external anal sphincters. One of them is under conscious control. Um, this is the histology of the large intestine. It doesn't have any villi. It's a flat surface without any villi. It contains goblet cells, which produce mucus, and it contains these deep intestinal glands present here. Large intestine is going to perform a reabsorption of water, vitamins, organic wastes, bile salts, and toxins. Um, or mm, Here we have a view of the defecation reflex. The defecation reflex is going to be initiated by distension of the rectum, and this acts upon stress receptors, which eventually activate more portions of the sympathetic system, which increase peristalsis, and eventually that leads to that uncomfortable feeling that's present whenever the need arises. This is a view of this is a high level view of the processing and absorption of nutrients, how at which levels uh, the digestion and absorption of these particular components happen. So let's look at carbohydrate digestion and absorption. It initiates in the mouth through salivary amylase, but also through brush border enzymes. Absorption occurs mostly across the intestinal epithelia. We mentioned this is mostly done in the jejunum. Lipid digestion, it uses lingual lipase and pancreatic lipase. Bile salts aid in this process through emulsification. Um, these lipid bile salt complexes form these micelles, which are smaller in size and aiding in and are easier to digest. These diffuse into the intestinal epithelia and eventually they mix with proteins and they form these chylomicrons. Proteins, the pH, the acidic pH destroyed the tertiary and quaternary structures of proteins, and then these are easier to access by these digestive these protein digestive enzymes. Absorption of water, nearly all is absorbed via osmosis, large portion of it in the large intestine. Ions, it's done through diffusion, co-transport, and active transport. And vitamins are absorbed by diffusion, and fat-soluble vitamins are absorbed as part of my cells. Here's a view of what happens through the secretion and absorption of water. Food and drink provides 250 milliliters. Saliva involves we release into the lumen 1500 millimeters. We have also gastric secretion, liver secretion, intestinal secretions, pancreatic juices, and colonic mucus. All of these add into the amount of water that is being released into daily into the digestive tract. Eventually, we also have the reabsorption to the small intestine and in large part to the large intestine until we lose very little water within our excretions. This is a view of the types of transports that different ions and vitamins occur and under what components regulated. All right, that's it for today. I hope there is a lot, the digestive system is rather complex, and we went through a lot of material. So if you need to review, follow this lecture alongside your book in order to dive deeper into some of the material. Thank you for your time.